Okay, thank you. So in some of the talks this morning, evolutionary rescue was already appearing. So what I want to do is tell a few sort of general concepts and then show applications to bacterial evolution. So to clarify once more, what is evolutionary rescue? What you can see here is the population size as a function of time. And prior to environmental change, the population is well adapted. So population size is stable, but then a change in the environment occurs, the population size declines, and the population is going extinct unless evolution rescues it. So it can happen that a rescue mutant appears in the population and the population size recovers. In that case, we are speaking of evolutionary rescue. In such scenarios, they are of course of fundamental interest in ecology and evolution, but they are also of great applied relevance in conservation biology, where we want that species adapt and survive, but also, as we've already heard this morning, in agriculture and in medicine, where the evolution of pesticide or drug resistance constitute instances of undesired evolutionary rescue. So what does the probability of rescue depend on? First of all, this rescue mutant needs to be present in the population either prior to environmental change, or it needs to appear de novo during population decline. However, that is not enough, because usually, especially if it appears de novo, it's only it's appearing in a single copy. If it's prior there in environmental, before the environment changes, it might also be just segregating at a very low frequency. And so this is a very stochastic process, because like even well-adapted individuals may just fail to leave offspring. And so even if it's appearing in the population, it's likely to get lost again by stochasticity. You can see this here. It was appearing 2 times, but it just got lost again. And only the 3rd time it appears, really the population got rescued. Okay, so how can we model evolutionary rescue? That's the most simple model one can possibly think of. There are just 2 types. There's a wild type and there's a rescue mutant. In the old environment, the population size is stable at some size n0. The mutant is deleterious and segregating in the population at some low frequency PM. In the new environment, the wild type has a birth rate lambda W and a death rate mu W, and the death rate is larger than the birth rate, so the net growth rate is negative. So the population size decays exponentially. By contrast, the mutant has a positive growth rate, SM, so in principle it can grow in this new environment. How do we now determine the probability of rescue, so whether the population goes extinct or not? So first of all, as I said, the rescue mutant needs to be somewhere in the, present in the population. Either it's already there, with this, like in this number PM times N0, that's the mutants we have at the time of environmental change, or it may appear de novo during population decline. And that's happening at a rate that's proportional to the mutation probability U, the rate at which the wild type replicates, and the total wild type population size. So we assume here now that the population size of the wild type, it's initially large, so we can just model the dynamics deterministically. It's an exponential decline. But as I said before, this is not enough. The mutant also has to escape stochastic loss. And that's just a simple birth-death process if you think of the mutants. And the probability that it establishes in the population and there's a permanent lineage rescuing the population, that is just 1 minus the probability of extinction of this birth-death process. It's a very well-known result, so the extinction probability is mu over lambda. Now we need to put the 2 components together. That's here, that's the probability of rescue is now 1 minus the probability that the population goes extinct. If you look at the second term first, this is that all the mutants that are there by the time the environment changes, that all of those go extinct, so none of them establishes. And the other term, that's the probability there's like that none of this rescue mutants appearing de novo. That's the zeroth term of a Poisson distribution. What you see in the exponent is basically the product of the rate of appearance of the mutation times the establishment probability, so the rate at which successful rescue mutants appear in the population. So now you can wonder, I mean, it was a very, very simple model. It's just a 2-type birth-death process. We could just calculate the probability of rescue just exactly analytically. So why this approach? And one reason is it can give us additional insight. I would like to show one small example for that. That's a tiny part of a big project that my former student, Christine Newton, did. It's about antibiotic resistance evolution, comparing bacteriostatic and bactericidal antibiotics. So antibiotics can be roughly divided into drugs that are bacteriostatic, so that they're decreasing the replication rate of the bacteria, or bactericidal, so they're killing bacteria. So here with this bacteriostatic drug, you can see that the birth rate of the bacteria is reduced by this factor 1 minus sigma, which depends on the antibiotic concentration C, and the death rate is unchanged. And there are two types of bactericidal drugs. The first one, so the birth rate is the same as the replication rate, but the death rate is increased. So this is a drug that's killing independent of replication. However, a lot of drugs actually don't kill cells that are metabolically inactive. And so we are, as an example of a drug that's actually just dividing, that's killing just dividing cells, we are looking at a drug here as an example that's killing the bacteria during cell division. So you have that basically the replication rate is reduced by a factor, but the cells that are not that are then killed are added at this death rate. And for the sake of comparison, we want to assume now that the net effect of the drug is the same for all of these three cases. So the net growth rate of the bacteria would be the same. So we can then reformulate or rewrite the birth and death rates by this just net effect E, which in the first type of drug, it goes to the replication rate, the second to the death rate, and turns out for this bactericidal drugs that's killing during cell replication, half of the effect is on reducing birth rate, half of the effect is on increasing the death rate. The graph here shows you the net growth rate as a function of the antibiotic concentration. And you can see like the growth rate goes down the larger the antibiotic concentration. It's true for the sensitive type in blue and also for the resistant type. It's just that the resistant type can withstand higher drug concentration. And this MIC, which you can also see in this figure, this is just a concentration where the net growth rate becomes negative. Okay, so now the question is like, is there a higher probability of successful treatment with a bacteriostatic or bactericidal drug? So what is now on the y-axis of this graph is the probability of treatment success. So this is a probability that there's no rescue and the population goes extinct. And this graph here is for no pre-existing resistance. And you can see here that for the bacteriostatic drug, we are having the highest probability of treatment success. Now we can ask, why is that? And for that exactly, it's helpful to look at these two factors. So this total number of mutants that are appearing during population decline, the mutational input, and the establishment of mutations if they appear. And you can see here, this bacteriostatic drug, it's leading to the lowest number of mutations, which is kind of clear because like if there's no replication happening, and we're assuming that mutations happen at cell replication, then there will be very few new mutations, but it actually has the highest establishment probability if a mutation appears. Nevertheless, it turns out, if we put it together, that this first factor, that's lower number of new mutation, is a dominant one. But if you think about it, you can already now kind of figure out what's going to happen when there's pre-existing resistance. And you can see here now, if the drug concentration is high enough, actually this bacteriostatic drug gets the worst option, and the drug that's killing independent of cell replication is the best treatment option, because that one has the lowest establishment probability, and that becomes just more important if you are already having bacteria-resident bacteria present at the time of treatment onset. So this is one of the advantages of this approach, that we can sort of understand better how do actually these results come together. The second is that the approach can be generalized, so we can't just look at this very simple model, but also at a more complex model. And the strategy is exactly the same. We look at the mutational input, so determining the initial number of rescue mutants in the population, then model the dynamics of the wild type deterministically. For this, we are getting then, from this, we are getting then the rate of de novo appearance of mutations. And then we look at the establishment probability of rescue mutant by branching process theory. And that can also be more complicated than the simple birth-death process. It can be time inhomogeneous, multi-type, and so on. And then it's again put together in the same way, where you have the zeroth term of a Porter distribution, and then also the extinction probability from all the mutants that are already present in the population. Because, yeah, there's not a lot of time. I will not show you an example for this general approach, but I would like to go back once more to antibiotic resistance evolution. So this is an instance of undesired evolutionary rescue, and yeah, it's one of the biggest threats to global health. And the question is, how can we manage evolution with smart treatment choices? Because like with the way we treat, we influences how the pathogens evolve, and we want to do this in a way that drug resistance ideally does not evolve, or at least slow down the evolution of drug resistance. So one very prominent approach are multi-drug treatment strategies, where one doesn't use just one drug, but two or more. And I mean, the idea is kind of simple. The idea is to increase the genetic barrier to resistance, because ideally the bacteria need at least one mutation to become resistant to each of the drug and the treatment. That can happen at the community level. So for example, if you're thinking of a hospital ward where we could have drug cycling, for example, 3 months, the default drug is drug A, and then 3 months it's B, and then it's going back to A and B, so on. Or mixing, where a fraction of people receives one drug and the other patients receive drug B. There are also multi-drug treatment strategies within patient, and the most straightforward, of course, combination therapy, where one gives 2 or more drugs simultaneously, or one could also cycle drugs during treatment of an individual patient. So what I want to focus on now is this combination therapy, so 2 or more drugs together. This is something that has been used for tuberculosis already for 80 years. It's not so much used for other bacterial infections. Another example, like in non-bacteria, is HIV, where this is, of course, a standard treatment. It's, as I said, it's sort of the idea behind it is simple. And I mean, there's these examples of tuberculosis and HIV where we know that this is a really good thing to do. But nevertheless, it's not clear whether it's always the best treatment option. And also, like, there's so many options of how to combine different drugs. So which drugs do we take? For example, like with the mode of action, what I showed before, how many drugs, 2, or do we need more? At which ratios, 50-50 or something uneven, and so on. And so my student Christine that you saw before on the slide, she really looked at an insane number of scenarios. I just want to show you one example here now, which is again coming back to this mode of action, bactericidal and bacteriostatic drugs. So now you're seeing here 3 different combination treatments. There's a one where this bactericidal drug that's killing independent of replication is combined with a static drug, or the 2 static drugs are combined, or 2 cidal drugs that kill independent of killing. You see this for 3 different mutation probabilities. It's easiest if you look at C. And there you can see that this mixed combination treatment with the drugs with a 2 different mode of action actually gives the highest probability of treatment success. If you go further to the left, you see then it's equally good as a CK plus CK treatment. We can again ask, why is that? And I put it here in a table, otherwise it's a lot of lines. If you look at the number of new mutants, actually the best strategy would be to use 2 bacteriostatic drugs, then we would have the lowest number of mutants appearing. However, that's the worst for the establishment probability, so that would have been. Now, if then resistance appears, the resistant bacteria would have the highest establishment probability. Then for the CK plus CK combination, it's opposite. And you can see that the one that's overall the best is actually medium in both of these components, the number of new mutants appearing and the establishment probability. Okay, so to sum up, evolutionary rescue is an umbrella for research about adaptive evolution to escape extinction. And the probability of rescue can be calculated by looking at those 2 factors. So the number of rescue mutants that are either present there already prior to environmental change, or that are generated de novo during population decline. And the second factor is the stochastic establishment probability. This sort of semi-deterministic approach, where we model the wild type deterministically and the establishment probability of this rescue mutant stochastically, this is sort of at least two advantages. One is this clear separation in this mutational input on the one hand and establishment probability that can often provide additional insight. And the second is that more or less complex scenarios can be analyzed in that way. And again, so resistance to antibiotics, also cancer chemotherapy, what we saw this morning, or pesticides, those all constitute instances of undesired evolutionary rescue. And so by modeling these scenarios, we can hopefully develop treatments based on evolutionary principles that prevent or slow down the evolution of resistance. Yeah, I would like to highlight Christine. So she did a lot of work on this multi-track treatment strategies, both combination therapy and sequential therapy. If you're interested in that, you should invite her. She's an excellent speaker. The project was a collaboration with Sebastian Bonhoeffer. And I also would like to thank the organizers. It's a really nice meeting, and thank you for your attention.
Go ahead and do one question for lunch.
I think it's
review that you talked about, not the review, but the classic model, right? You have kind of this death processing of the two, presumably for the and if you use branching process, you can make it more generalized, like the main final. But I'm wondering, like some of the talks earlier this morning, that establishment probability does not depend on the number of kind of wild type or kind of original population size, right? There could be competition.
Yeah, you can put this in actually. I mean, so because what the branching process assumption, the only thing it assumes is that the mutants reproduce independent from each other. But when put in the sort of wild type population size and the competition coming from this wild types, and then it's just a time inhomogeneous process. So that works. If you want to know about like interactions between mutants for establishment, this is something that there's a discussion session this afternoon about, where I hope that maybe people have some ideas on that. Yeah, but like this competition can be built into the branching process. Yeah.